Hi, my name is Kim Woo-jin. My subject for URP is secondhand smoke increases pCO2 in animals' blood and may cause acid-base disorders. I studied how secondhand smoke affects animals in terms of acid-base equilibrium. First, I want to talk about why I chose the topic related to secondhand smoke. Nowadays, the harmfulness of secondhand smoke is widely recognized throughout society. However, I felt that there was still a lack of awareness about the effects of secondhand smoke on animals. Humans are not the only ones exposed to secondhand smoke. Companion animals and livestock living with humans can also be exposed to secondhand smoke. According to Animal and Plant Quarantine Agency, almost 6 million households in Korea have companion animals. Especially, companion animals living with cities will have more chance to be exposed by secondhand smoke. So I felt the need to study the effects of secondhand smoke on animals. More specifically, I studied about how secondhand smoke affects rats' blood pCO2 and acid-base equilibrium, and I checked if there was any chance of causing acid-base disorder. My hypothesis, like the subject of my research, is secondhand smoke will increase pCO2 in animals' blood and may cause acid-base disorders. I thought cigarette smoke would interfere with the exchange of gas in the lungs. If, as I expected, the exchange of gas in the lungs is interrupted by smoke, pCO2 in the blood will increase as shown in the graph at the left. As we all learned in biochemistry class, if the pCO2 in the blood increases, the buffer reaction will proceed to the right in the blood, and the pH of the blood will be decreased by the protonation of carbonic acid. And if this condition persists or worsens, it could cause acid-base disorders such as respiratory acidosis. To prove my hypothesis, I designed the experiment as shown. If the experiment showed a significant increase in pCO2 of mouse blood, we could say the hypothesis is proved. From now on, I will explain details about materials and methods. In this experiment, I used 10-12 weeks old preg rats. Those weigh between 250 grams to 255 grams. I randomly divided them into two groups of five. One group for second-hand smoke exposure, which I'll call an experimental group from now on, and one group for control group to compare with experimental group. They were anesthetized by muscle injection of mixture of ketamine HCl and xylene HCl during the whole process of experiment. After animals were anesthetized, they were put into the acrylic box called smoke exposure chamber for secondhand smoke exposure. This acrylic chamber was specially designed for this experiment. We will discuss about its detail later. Before putting rats into the chamber, cigarette was lit and set to the chamber first, and after waiting for the cigarette smoke to fill the chamber, rats were placed inside, and let them inhale cigarette smoke naturally for 15 minutes. For control group, they were anesthetized and placed at the chamber for 15 minutes as experimental group, but without exposing them to cigarette smoke. After the process, 1.2 cc of arterial blood was collected from carotid artery of all experimental and control group for blood test. Blood test was conducted by arterial blood gas analysis, also known as ABGA. This is the detail of structure of smoke exposure chamber used to perform second-hand smoke exposure environment. The chamber was made of transparent acrylic panels so that the experimenter can check the internal situation. Chamber is 15 cm wide, 15 cm high, and 40 centimeter long and has 13.5 centimeter high wall on both sides to prevent rats from directly touching the cigarette. The distance between those walls is 30 centimeters, and three holes were punched at posterior wall at 5 centimeter high from bottom for air ventilation. Cigarettes were installed leftmost hole among those holes. And the cigarettes used in this experiment were domestic cigarettes with filters, which their ingredients were disclosed, such as nicotine content. As a result of ABGA after the exposure to smoke, both PO2 and pCO2 in experimental group has significantly increased. All values of the experiment are expressed as mean plus minus standard deviations. The statistical significance was tested by ANOVA and students t-test to determine p-value under 0.05 as significant. After the second-hand smoke exposure, pCO2 of arterial blood of experimental group has significantly increased 56 to 0.2 plus minus 8.1 millimeter of mercury to 66.3 plus minus 10.6 millimeters of mercury. Actually, I expected that pCO2 would decrease after the exposure. However, the results shown an increase in pCO2 after the experiment. This is not a result that has a significant impact on hypothesis testing, but further study is expected to be required as to what factor influenced this result. The case of pCO2 of arterial blood of experimental group after the second-hand smoke exposure, there were significant increase of 46.0 plus minus 6.3 to 53.5 plus minus 2.7 millimeters of mercury as expected before experiment. Table shown left is the normative data of ABGA result of anesthetized rat. Comparing it to the result, the pCO2 of experimental group is contained within the range of suggested inclusion criteria, but is very close to the upper limit and appears to be quite high compared to the average. And it's even slightly beyond upper limits if comparing it to criteria of 25th to 75th quartile range. Based on the above results, these results can be determined to be fairly high and could be analyzed to be sufficient to bring acid-base imbalances. ABGA analysis in the acute second-hand smoke model demonstrated a significant increase in blood carbon dioxide partial pressure after second-hand smoke from 46.0 plus minus 6.3 millimeter of mercury to 53.5 plus minus 2.7 millimeters of mercury. Comparing this result to normative data, it seems sufficient to determine pCO2 of experimental group is abnormally high and could cause acid-base imbalance. An ultimate cause of this abnormal status seems to be due to smoke interfering with gas exchange in the lungs. If the cause of the increase in pCO2 persists, the acid-base imbalance of blood may persist and cause respiratory acidosis. In summing up above, it can be concluded that second-hand smoking increases pCO2 of animals' blood and may cause acid-base disorders. Comparison of pCO2 concentration between the experimental group and the control group through the ABGA demonstrated that second-hand smoke interfered with gas exchange in animals' respiration and caused imbalance of pH constancy. This suggests that continuous exposure to second-hand smoke is likely to cause acid-base disorders such as respiratory acidosis. This is the conclusion of my study about relation between second-hand smoke and animals' acid-base disorders. That's all for my research presentation, and I thank you very much for hearing.